Hello everyone, thank you for attending this talk. The study presented here is part of a cooperative research and development agreement with EPA ORD project lead Dr. Jim Goodrich, whom is a co-author in this study, Washington State University and Aquasense Technologies. The Electric Power Research Institute and Black & Beach Consulting were also collaborators in this study. Legionellosis is a general term describing all bacterial infections caused by Legionella. The figure on the left is a plot of case numbers in blue and incident rate in orange per 100,000 people since 1976, when the large outbreak occurred during the American Legion Convention and infections became a notifiable disease. Since 1984, the incident rate has increased more than sevenfold, with case numbers starting at 235 from the 1976 convention and increasing to about 10,000 annually in 2018. So why this large increase? Possible explanations are disease awareness and increased environmental and clinical testing, aging water infrastructure, and other significant changes in water quality within premise plumbing systems, including water conservation efforts, increases in susceptible populations, and underlying illnesses, climate change, and other weather, air quality, watershed-related influences on Legionella bacteria prevalence and exposure risk. For the graph on the right, the first set of bars shows the percentage of outbreaks by environmental source, with exposure to potable water, white bar, making up the majority of these outbreak cases. However, in the second set of bars, indicating total confirmed and suspected cases, the environmental source is almost evenly split between potable water and cooling tower water exposure. From 2014 data documenting emergency department visits and hospitalizations, the estimated healthcare cost for Legionella cases was about $400 million. This bacterium is a significant public health and economic burden. Currently, there are over 60 species or members of the Legionella genus. About a third of these species have been associated with human disease. Legionella pneumophila, or LP, is responsible for most documented cases. Within the LP species, there are 15 serogroups, and they are identified as belonging to a particular serogroup using a panel of antibodies. I will explain this more in the next slide. LP serogroup 1 types make up more than 80% of those isolated from clinical cases. The left figure is a graphical depiction of the Legionella cell membrane. On the outer membrane, there are long chains of lipopolysaccharides, or LPS molecules, covering the membrane surface, which are shown as chains of orange and red hexagons. The right figure graphically depicts one of these LPS molecules. The antibodies I mentioned in the previous slide that determine the LP serogroup recognizes the top portion of the LPS molecule, or the O-specific chain, shown as the red hexagons. This portion is unique to each LP serogroup and is what the panel of antibodies are recognizing. Thus, when you hear LP serogroup 1 or 4 or 6, they are the same species, but they have slightly different LPS structures. Legionella are ubiquitous in the environment, especially in water and soil, and unfortunately, the conditions found within premise plumbing or engineered water systems are conducive for their growth and higher exposure risks to them because of the generation of breathable drinking water-derived aerosols from showers, decorative features, water misters, cooling towers, and so on, presence of dead legs and cross connections, high surface area to volume ratios, presence of eukaryotic hosts that are susceptible to and can amplify harmful bacteria. Water can stagnate in these systems, further degrading its quality. There is low to absent disinfectant residual, amenable growth temperatures and gradients due to hot water systems, presence of biofilms and sediments, all of which comes together to form a nutrient-rich and conducive microbial growth environment like an incubator. One of the primary goals in this EPA research area is to prevent pathogens from colonizing our water systems, and we can do that by determining the operational, engineering, and water quality parameters that lead to pathogen occurrence and amplification. So those conditions can be managed or avoided. And we also evaluate technology or methodologies to decontaminate these systems. To give additional background information for this study, the figure on the left shows the electromagnetic spectrum, with the visible light range shown as the rainbow band in the middle. The ultraviolet or beyond violet range is expanded on the right side into UVA, UVB, and UVC wavelength. What separates UV from the visible spectrum is its ability to induce chemical reactions and ionize molecules. This figure shows the different forms of DNA nucleotide dimerization according to type of UV light inducing them. These dimerized nucleotide sites can be referred to as DNA lesions and basically causes kinks in the classic double helix structure of the DNA, which can then negatively impact DNA replication, transcription, and ultimately cellular function. UV radiation in the UVC range, or the 200 to 80 nanometers, is germicidal, with UV disinfection being a recognized and proven technology for decreasing waterborne disease risk from microbial pathogens. Low-pressure mercury arc lamps are commonly used in drinking water and wastewater treatment plants. UV light-emitting diodes, or LEDs, are an alternative method of UV delivery, as these LEDs can also emit radiation at germicidal wavelengths. This slide summarizes several studies evaluating UV inactivation of Legionella. More studies have been conducted using low-pressure mercury lamps. There have been more Legionella strains tested, and there are a range of fluences reported that results in 2 to 4 log reductions of Legionella. Fluence is the more accurate term for UV dose and is expressed here as millijoules per square centimeter. Less studies with fewer strains tested have been published detailing UVC LED inactivation, especially in drinking water matrices and modeled premise plumbing systems. Thus, the study presented in this talk hopes to fill those knowledge gaps. To do this, we use the AquaSense Pearl Beam collimated beam device to evaluate UVC LED inactivation of various Legionella pneumophila serogroups at 3 wavelengths and several fluences. Samples are typically placed in a petri dish under the UV light generated from this column. Based on the size of the petri dish, volume of fluid in the dish, percent UV transmission or UVT of the test medium, and target fluence, you can determine the exposure time for the test suspension to achieve that fluence or UV dose, which is listed in the table here. For each of the 3 wavelengths shown in the columns, 255, 265, and 280 nanometers, several fluences were evaluated and are listed as rows. 0.5, 1, 2, 5, and 10 fluents were tested, with an additional 16 and 34 fluents evaluated at 280 nanometers, which I will explain why later. For our study, we tested 4 Legionella pneumophila strains, 2 serogroup 1s, one isolated from human samples, termed clinical isolate, another from drinking water. We tested a serogroup 4 clinical strain and a serogroup 6 clinical strain. Each fluence was tested 3 times for each strain at each wavelength. These are the results at 255 nanometers, with the blue lines indicating log CFU levels on the y-axis after exposure to 0.5, 1, 2, 5, and 10 fluences along the x-axis. Starting levels were between 6.4 to 6.8 logs of CFU per mil. There were 1.6 to 2 log decreases in CFU at 0.5 fluents, or after 17 seconds of exposure, shown here by the red arrows, and a larger spread of decreases at 1 fluence, or 34 seconds of exposure, shown by the green arrows. However, at a fluence of 2, a 1 minute 7 second exposure, highlighted by the blue arrows, the serogroup 1 isolate, the drinking water one in particular, were the only ones with detectable CFU levels. Overall, all strains were inactivated fairly quickly at this wavelength. The green lines show the log CFU levels after exposure at the 265 nanometer wavelength, superimposed on the data from the previous slide. Similarly, there were slight differences in the log reductions between each strain and fluence, but statistically, the significant differences were between the serogroup 1 and serogroup 4 isolates, which were less susceptible compared to the serogroup 1 drinking water and serogroup 6 isolate to inactivation at 5 fluents. The orange line represents the log CFU levels measured at 280 nanometers. The only change from the previous graphs is the expansion of the x-axis to include the higher fluences evaluated at this wavelength. The serogroup 1 drinking water isolate had the steepest line, indicating it was more quickly inactivated, followed by serogroup 6, shown below, and serogroup 1 and 4, which were both the least susceptible to inactivation. As I mentioned earlier, for this wavelength, the additional 16 and 34 millijoules per square centimeter fluences were added to the test because a point of entry UV treatment device was tested next. This device operates with a 280 nanometer LED, and at the tap water flow rates of our test setup, it was estimated that the fluence would be near the 16 to 34 range tested here with a collimated beam. This is our point of entry setup under a biological safety cabinet. Moving from left to right, the tap water source is delivered via Decon tubing connected to the lab sink faucet. There is a flow meter to monitor the rate. For all experiments, we observed between 2.5 to 3 gallons per minute flow with this setup. There was a sampling port to collect and measure the water quality for each experiment. We used a pump to deliver the Legionella pneumophila inoculum into the system, which was followed by a stretch of a pipe segment with a static mixer. There is a pre-treatment sample port to measure the Legionella levels prior to UV exposure and after mixing into the tap water. After the Legionella contaminated water enters the UV unit, there's another sampling port to collect the UV treated water, while the remaining water is diverted to a 55-gallon drum serving as a biological liquid waste collector. The same 4 strains were tested. The images on the top left are of my EPA colleagues who helped with the experimental runs. John Hall, who is a co-author in this study, is next to the waste drum, and Dr. Jeff Szabo is in front of the sink, which was the source of the tap water. I'm in the blue lab coat. My other co-author, Dr. Jim Goodrich, is behind the camera. For each strain tested, we performed 5 runs or replicates. After collecting the tap water from the sample port by the flow meter for background water quality measurements, in this exact sequence, the tap water was turned on, then the Deca unit, since it requires a water flow for proper operation, then the pump was turned on at a predetermined flow rate to feed the concentrated Legionella inoculum into the tap water to achieve the desired test concentration. Then we manually released the Legionella inoculum tube clamp. We waited about 5 seconds, then collected 1 liter at the pre-treatment port below the UV unit, which took about half a minute. Then 1 liter was collected at the post-treatment port above the UV unit. We then engaged the Legionella tube clamp to avoid backflow into the bacterial stock flask. We turned off the pump, and we flushed the entire system, including the pre- and post-treatment sampling ports. The UV unit was then turned off, followed by the tap water. Then we repeated this process 4 more times. It should be emphasized that this UV point of entry unit was not designed for this kind of setup, but we needed to test the system systematically. The top table indicates the water quality parameters required for optimal performance of the test unit as stated in the product manual. The bottom table shows the water quality parameters measured during the test runs for each strain. In Cincinnati, we have very high pH and hard water, so these measurements were expected. The water temperature was between 10 to 20 degrees C, but was lower by about 6 to 10 degrees C during the Serogroup 1 clinical strain test run. The free and total chlorine levels were consistent, but was lower during the Serogroup 4 run. It is ideal to have similar water quality conditions for comparisons between each strain, and although these experiments were all conducted within a 1-month time frame, this just illustrates the variability in water quality, which we all know can fluctuate wildly even within a single day. This is a plot of log reductions calculated by subtracting the post-UV treated CFU levels from the pre-treatment levels. Values are listed in the table on the right. The results for the Serogroup 1 drinking water isolate were surprising. Based on the collimated beam experiments, we expected this isolate to have the highest log reductions, but here it displayed the lowest. We expected higher log reductions for the Serogroup 6 strain, which we did observe here at about 5. However, the Serogroup 1 and 4 clinical isolates was expected to display lower inactivation levels compared to the other 2 strains, but in this case, the log reductions were only lower compared to the Serogroup 6 and not the Serogroup 1 drinking water isolate. Overall, these differences were very interesting because at the beginning of my talk, I mentioned that the LPS structure were what differentiated these strains, and most of the genome should be very similar since they are all members of the same species. The primary mechanism of action for UVC, especially at the 280 wavelength, is thought to be dimerization of nucleotides in the bacterial DNA. However, this data might suggest that other molecules like lipids and proteins may be targeted under these conditions as well. With the free chlorine levels at 0.7 to 0.9 ppm during these runs, along with the UVC LED unit, I expected high inactivation levels. However, this data suggests that pathogen inactivation may be reduced in the presence of the chemical and microbiological background commonly present in tap water. Efficacy of UV inactivation can differ even between strains of the same species. Understanding these differences could be useful, especially in cases where an environmental source is linked to a clinical case and an effective remediation strategy is needed to target that specific isolate. In this study simulating high contaminant loads, the point of entry DECA device demonstrated at least a 3 log upwards to 5 log reduction of Legionella. Further work should aim to optimize the operating conditions and setup of this device for complete inactivation, and we would also like to explore the inactivation of biofilm-associated forms of pathogens that may be inactivated differently or less efficiently than their evenly suspended or surface-attached forms using this technology. Thank you for your time.